Good morning, everyone. We are starting a new chapter this week, today. We'll be looking at the cytoskeleton of the cell. Uh, Everyone should have been able to view their quiz responses and see the correct answer. Our exam on Wednesday, we had an average of 70%. Not quite as good as this last exam, but this was probably the hardest collection of material for the course. Um, we still have a pretty good average for the entire course, including all of the reef points of 77%, and I hope we'll be able to stay with that average for our course. I haven't had too many people ask questions about the exams, but so if you do have any, feel free to email me if you have a specific question, you don't understand why your response was wrong, you want some clarification on that material. Because remember that our final exam will be comprehensive, so you'll be tested on all of the material that we've learned during this semester. So today, starting with something new, make sure that you have signed into your Reef account, that you have joined our course, and we will look at different components of the cytoskeleton. We actually see examples of a couple of these different three categories of cytoskeleton, and we'll look at each of them in order. In our figure here, we see an animal cell. And of course, cytoskeletal components are present in all eukaryotic cells, but we will be primarily looking at the different components and their roles in animal cells, one of which we see here. Here we have our nucleus that's stained blue, and we have fluorescently labeled both our microtubules here in green. Notice how they're extending from sort of the central portion of the cell and extending out towards the periphery. And in red, we have stained actin filaments that notice sort of form a structure around the periphery, but also extend out in this animal cell that looks like it's sort of moving, it's migrating towards the right of the image. So we'll look at not only the structure, but the function of these different types of filaments that are present in all cells, all eukaryotic cells. As a definition, it's a network of protein filaments carrying out very many different types of roles in the cell, supporting the cell, allowing for movement, allowing cells to adhere to each other, have roles in cell division as well. We divide them into three categories. The largest filaments are microtubules, largest in diameter, about 25 nanometers in diameter. Actin filaments are the thinnest filaments in the cytoskeleton. They are about 7 nanometers in diameter. So those are the thin ones. And the category of filaments that are intermediate in size, we put in this class of intermediate filaments because it's actually a large family of different types of filaments that make up this group. And that's what we're going to talk about first. Our intermediate filaments. These are the strongest of all of the types of filaments that are part of the cytoskeleton. These cable-like structures, about 10 nanometers in diameter. We will find them throughout may find them throughout the cytoplasm of a animal cell. Notice in some cases they actually can extend across the length or the width of this cell. We find them, if we look down, we find them in epithelial layers of cells. If we look down on a sheet of epithelial cells, we see what's labeled in green are these intermediate filaments extending out towards the periphery. And we can see that in some places, it looks like the intermediate filaments of one cell are in contact with the intermediate filaments of another cell. This is one way that cells will be linked to each other through a junction that we call a desmosome. And that's what we see on this drawing here. We'll look at desmosomes in a little bit more detail a little bit later. So we have this very strong, high tensile strength cable-like fiber extending through the cells, primarily epithelial cells. And there's other examples. Here we're looking at keratin, which is the name of the intermediate filament that we find in epithelia. These are sheets of cells that have to function together. The cells all work together. So what makes up this cable? What are the individual polypeptides that together form this filament? Again, intermediate filaments are a large family of filaments composed of different types of polypeptides, but each of those polypeptides always as a subunit, as a monomer, have a characteristic shape. Long helical, alpha helical rod central portion, and the amino terminus and the carboxy terminus both have a globular structure. What we're going to see is how they assemble these individual monomers, these individual subunits, will assemble into this rope-like filament. The function in general, and we'll talk about some specifics, is to enable cells to withstand mechanical stress. These filaments will provide strength to a cell, tensile strength, their ability to resist or withstand mechanical stress onto the cell itself or to a sheet of cells, as we'll see. So what does that individual subunit do? What we'll see is that the subunits will assemble in a particular way. We have one monomer, we have our rod-like alpha helical region that will assemble with another monomer to form what we call a coiled dimer. Right? They'll coil around each other. Those dimers will then assemble together to form a tetramer. Notice that the tetramers, one dimer is oriented in the opposite direction to another dimer, with their amino terminus facing the ends. And then these tetramers will assemble with another tetramer in this stereotypical arrangement. And that's essentially one fiber of this filament. The actual cable-like structure, this rope-like filament, forms when 8 of these fibers assemble together. Here we've laid them out flat so we can see them, but they twist around each other to form this very strong rope-like cable structure. All of those globular heads and tails, and we'll think about them when we look at the function of this structure. They will be able to interact with other components of the cell. So think of them sort of as sticking out and being sticky, allowing this to interact with other proteins, other components of the cell. So what's one of the functions? Well, in epithelial cells, and remember what epithelial cells are assemble in a sheet. So an epithelium is a sheet of cells that are attached to each other, and the sheet of cells function sort of like the epithelium that lines the outer surface of our body, the inner lining of all of our organs. All of the cells have to act together as a sheet. Those cells will have keratin, is the type of intermediate filament that will be expressed and will assemble within the cytoplasm of those cells. And it serves as resistance to mechanical stress. If that sheet of cells has a tension applied or pressure applied, the cells will remain intact. In the absence of intermediate filaments that we see on the right-hand side, if this sheet of cells is stretched or has a pressure applied, the cells will not be able to stay together. They will continue stretching until they rupture. Now, each individual intermediate filament does not actually extend into its neighboring cell. What we see is they are associated with the neighboring cell at these junctions, these little black dots. Those junctions we call desmosomes. So the desmosome is a junction, a collection of proteins that allow the cytoskeleton of one cell to be connected to the cytoskeleton of a neighboring cell so that all of those epithelial cells can interact with each other. So if we're talking about epithelial cells, we're talking about keratins. We can see an example of this desmosome on this slide. So here is an electron micrograph showing the junction between this cell on the left-hand side that is attached to the cell on the right-hand side. And all of those components that we see are shown in our cartoon. Here in blue are our keratin filaments that are binding to this red plaque of anchor proteins. Right, so here's our cell on the left, here's our cell on the right. The cytoskeletal keratins are within each of these neighboring cells, but each cell, the intermediate filaments, the keratin, is bound to our red anchoring proteins. And those anchoring proteins are linked to our green transmembrane protein that we call a cadherin. Cadherin means calcium binding adhering proteins. So the cells are adhering to each other because all those cadherins will adhere to each other, will bind to each other. And each cadherin is attached through the membrane, transmembrane protein, to the anchor on either side. And so this whole complex we call our desmosome. How cells stay together and essentially spread the tension on one cell is spread onto the other cells so that all the cells can act together as a sheet. Now, those epithelial cells also are attached to the basal surface. So if we looked at the bottom of each of those cells, the basal side of those epithelial cells, we would see that they are all connected to this extracellular matrix that we call the basal lamina. And the cells, via their keratin filaments, are attached to or bound to that basal lamina through, again, anchoring proteins that are linked to a different type of transmembrane protein that we call integrin molecules. So not only are the epithelial cells attached to neighboring cells by desmosomes on the side, they're all attached to the same extracellular matrix. Extracellular matrix is a network of proteins that are secreted to the outside of the cells. On the basal side, we call it the basal lamina. Right, so the cells are attached to each other. They're all attached to this extracellular matrix through integrin molecules as well. This allows them to function together as a sheet. So I thought I would bring up one of our, whoops, and we'll go to an experiment. One of the SmartWork videos that shows us some of our, both the function and the assembly of intermediate filaments. So let me close that down for a second. Open up this guy. Let me change my audio. We can watch part of this.
Eukaryotic cells contain a complex network of filaments. intermediate filaments, microtubules, and actin filaments that provide the cells with strength, structure, and movement. Although all eukaryotic cells contain microtubules and actin filaments, intermediate filaments are found only in vertebrates and a number of other soft-bodied animals. Intermediate filaments are found in animal cells that require a lot of strength, such as the epithelial cells of the skin. Some of these filaments span the length of the cell, connecting cell-cell junctions called desmosomes. These cables of intermediate filaments have a high tensile strength. Without these filaments, stretching or pressure on the epithelial sheet would cause it to rupture. Each filament is rope-like, consisting of 8 thinner strands made of a precise hierarchical arrangement of protein subunits. At the lowest level, 2 monomers associate with each other to create a twisted dimer.
All right, we can stop here because we've discussed all of this, and you can watch the rest of it yourself if you have SmartBook. So, let me get us back. And
hold on a second, we should be back. All right, so let's look at an example of what happens if an intermediate filament does not form properly. And that's what this experiment is here. What they've done here is to demonstrate the importance or the role of keratins, intermediate filaments, in epithelium by injecting into a zygote, a fertilized egg, inject a plasmid that has a gene that is mutant, right? So a gene encoding an abnormal keratin monomer. Inject that into embryos, transfer the embryos into a foster mother, and if any of those embryos actually took up that plasmid, which is then expressing a mutant form of the keratin, we end up over here with a transgenic mouse. What we see as a phenotype of this transgenic mouse is its skin is blistered. This phenotype arises from the fact that even though all of the, this mouse, as well as all of the other mice, are expressing normal keratin subunit polypeptides, the presence of an abnormal subunit prevents all of the subunits from assembling properly to form that strong rope-like cable. So let's look at what is this skin blistering and what's causing it. Right? Here we're looking at the epidermis, the one of the layers of epithelial cells that make up the epidermis, the outer layer of skin. And they're showing our keratin as with these little dotted lines showing that the keratin has not formed properly. And so the keratin is a weak filament, not that strong cable. And because of that, even though, yes, the keratin's trying to bind to the desmosomes, connecting the cells to each other, binding to the hemidesmosomes, connecting the cell, holding the cells in place on the basal lamina, because they're weak, any pressure, any rubbing of this layer of cells will cause rupturing. Rupturing between the nuclear portion of the cell and the hemidesmosomes. The weakest portion of the cell will be ruptured. The cells will break. That will lead to the blistering. Can you people be careful? We've got a bunch of people chatting. Can you make sure that you're muted? Thank you. And so what happens then if, so here in this diagram, if we actually look at, and again, an electron micrograph of a section through skin, and our skin is composed of both epidermis, which is multiple layers of epithelial cells. The outer layers are actually dead cells that protect us from the environment, right? Chemical damage from, or physical damage from the environment. But the epidermis is our layers of cells on top of a basal lamina that we're pointing to with that arrow. The sort of inner layer of skin we call the dermis. And so what we're seeing is in normal skin, we have these multiple layers of epidermis, but in someone who has a disorder of defective keratin filaments, any type of rubbing or minor pressure on the skin is going to cause breakage of those cells that are attached to this basal lamina. Those cells will be disrupted, forming blisters. We can see this in someone who has inherited a disorder of keratin, which we call epidermal lysis, right? Lysis of the epidermis. Bullosa means blistering. So a genetic disorder due to mutations in keratin genes usually is manifested in young children, primarily feet and hands that are subjected to more rubbing due to extensive blistering due to any kind of pressure or minor trauma to the skin. Now, keratins are just one type of intermediate filament. Those are the filaments that are present in epithelial cells. Other types of intermediate filaments are present in connective tissue, in muscle cells, glial cells. These are cells, supportive cells of the nervous system. We call them vimentin and vimentin-related filaments. Nerve cells have a particular type of intermediate filaments that we call neurofilaments. And all cells have intermediate filaments that we call lamins. These are the intermediate filaments that line the inside and support the shape of the nucleus. So let's look at an example of first our neurofilaments, and then we'll remind ourselves of the role of lamins in the nucleus. Before we do that, let's see what we know about intermediate filaments right now. What can you tell me about structure and function of our intermediate filaments? So you're going to read all of these 4 statements and identify which statement is false. About 5 more seconds. All right, let me close it up. Now I see that everyone was following the leader, the first person who came up with an idea. But what about D? Each filament is 10 nanometers in diameter, right? a cell is only about 20 to 25 microns in diameter. So the filaments must be significantly smaller. That's the statement that's incorrect. Intermediate filaments can be found in the cytoplasm. We looked at examples of keratins. They can be found in the nucleus. Those are lamins. They can be anchored to the plasma membrane. That's what we saw in the example of keratins in epithelial. The individual polypeptides are a monomer, right? And covalent bonds link amino acids to each other to form a polypeptide. But polypeptides interact with other components, other polypeptides, by non-covalent bonding, right? They're attracted to each other by hydrogen bonds or electrostatic interactions, right? Relatively weak bonds, but there's so many of them that they're held together. So the individual components of an intermediate filament are held together by non-covalent bonds. So A, B, and C are all correct. Right, so this is just kind of a reminder, make sure that you're reading carefully when you have choices in multiple choice questions. Okay. 10 in diameter, correct. 10 what, right? 10 nanometers, that's a different unit than micrometers. So what about a different type of intermediate filament? Neurofilaments are what we call a different polypeptide that assembles in a similar way to form a different type of intermediate filament. These will arrange themselves in parallel arrays. They are cross-linked by cross bridges, and those would be either, that could be covalent bonding, holding individual filaments next to individual filaments, and sometimes involves other proteins. This is going to give axons mechanical strength. Axons can be very, very long, and so the presence of neurofilaments gives them some structure and support. If we look at a cross section of an axon, we can see all these tiny little dots are all of the neurofilaments that are running the length of that axon to give it some support. Not to be confused with a different type of cytoskeletal filament, large, as we can see, much larger in diameter, those microtubules that we'll be talking about next. Microtubules in an axon are going to form tracks or serve as tracks that vesicles full of neurotransmitter can travel from the nucleus down to the axon terminal. Right, so the neurofilaments are providing support and strength, these rope-like cables in axons, similar to, though carrying out a different role than the keratins in epithelial cells. And another type of intermediate filament is the lamins that are present in the nucleus. We've seen this image before, a mesh-like two-dimensional network of fibers on the inner face of the nuclear envelope. The nuclear lamina, shown here in our blue, lining the inner face of the nuclear envelope, is not only attached to the inner nuclear membrane, but is also attached to proteins of the chromatin. When lamins are phosphorylated, they will have weakened binding between the tetramers and the dimers that make up that rope-like structure, which causes the lamins to disassemble. When the lamins disassemble, that allows the nuclear envelope to fall apart at the beginning of meiosis as well. In order for a cell to divide, in order for those chromosomes to be separated, right, to opposite poles of a dividing cell, the nuclear envelope has to fall apart. Then later, when new daughter nuclei are forming in the daughter cells, once cell division has been completed, the lamins will be dephosphorylated, and that will allow the lamin fibers to reassemble the Lyman filaments, which will then allow them to essentially bring together all the different pieces of the nuclear envelope to form those daughter nuclei. So, question for you, thinking about nuclear lamins, right? The nuclear form of a particular type of intermediate filament. You add a drug to cells that are undergoing mitosis that inhibits the activity of an enzyme that dephosphorylates the lamins. What's going to happen to those cells? All right, so let's think about what do we know about mitosis? What do we just describe about nuclear lamins? 5 more seconds. All right, I'm going to close it up. Closing up here. And what we'd expect to happen is that the cells will be arrested in mitosis. Arrested means it will stop. It will come to a stop because the lamins, when they're phosphorylated, the cell will be able to phosphorylate its lamins and it will be able to initiate mitosis. But if the lamins cannot be dephosphorylated, then the cell will be stuck in the middle of mitosis. It won't be able to complete telophase. It won't be able to complete cytokinesis. It will be stuck in the middle of mitosis, right? The inability to dephosphorylate won't have anything to do with linking the chromatin to the nuclear envelope, won't have anything to do with the nuclei interacting with the plasma membrane. They never do that. Right, so that this is what we're talking about, and we're getting ready to actually talk about the cell cycle and regulation of the cell cycle a couple of chapters from now, where affecting the assembly of nuclear lamins is one of the roles of some proteins that regulate the cell cycle, the cell moving from interphase into mitosis and proceeding through mitosis. So let's look at another component of our cytoskeleton, our microtubules. Now these are the largest component of the cytoskeleton of the 3 different categories. And a microtubule is a microtubule. Every microtubule is made of the same subunit, a dimer, a tubulin dimer. There's an alpha subunit and a beta subunit. These cylinders, these hollow cylinders, are usually extending from a central region of the cell called the centrosome. We've heard of that when we think about cell division. The centrosome is the microtubule organizing center, where they start to be assembled, and they're growing from the center out towards the periphery of the cell. Many different functions for microtubules, generally not so much in supporting the cell, but more for organizing the different components inside the cell and allowing for movement either within the cell or allowing the cell itself to move. So let's look first at the structure of our microtubules. Again, the largest in diameter, 25 nanometers wide. Here's our little our little key, but 25 nanometer wide hollow cylinder. Growing from our centrosome, which is usually close to our nucleus, extending and growing out towards the periphery. We can see that in an interphase cell. We also can see the activity or the role for microtubules in a dividing cell. That centrosome would have divided during interphase. The centrosomes would then move to the poles of this dividing cell. Individual microtubules are assembling and growing out from the centrosome, some of them growing towards the center, the midline of that cell, where they'll interact with chromosomes, right? So these microtubules have to be dynamic. They have to be able to grow, and then they have to shrink in order to move those chromosomes around during mitosis. In some cell types, microtubules have even a different role. In cells that have cilia extensions of the plasma membrane, the microtubules allow for movement of that cilia, that oar-like motion, beating motion of cilia that can either allow for fluids to move past the cell, or if this was a unicellular organism, the beating of cilia would allow to propel that cell through its liquid environment, its aqueous environment. So the first thing we're going to look at is, what are these microtubules made up of? Notice we have dark green and light green components, globular proteins, alpha and beta subunits that will assemble to form our 25 nanometer hollow cylinder. The cylinder is composed of these, what we call protofilaments, 13 filamentous arrangements of our tubulin heterodimer. We call it a heterodimer because it's a dimer, two polypeptides associated with each other. They're not identical. One we call a beta subunit, one we call an alpha subunit. Very similar, but they're different polypeptides. That heterodimer is a subunit. Heterodimers will be added to what we call the plus end of the microtubule. It would have initiated at this end. It's going to be growing at its plus end. It gets longer at its plus end. It can also get shorter at its plus end. So our dimer consists of an alpha tubulin and a beta tubulin. Each of those tubulin polypeptides can bind GTP, and they're polarized. The beta tubulin is the plus component. It is at the growing end. So how are the microtubules changing their shape? Again, they are dynamic. They are changing all of the time. Dimers are added one at a time to the plus end of the microtubule. The dimers, when they're added to a growing microtubule, are bound to GTP. But we know any GTP binding protein also can hydrolyze that bound GTP to GDP, and that will affect how the heterodimers associate with each other. So let's look at an example of the dynamic nature of these microtubules. We sort of turn them on our side here, right? This would be our minus end here, and here's the plus end, the growing end. What our figure is showing us is our beta and our alpha tubulin of our tubulin heterodimer are both bound to GTP, right? And that's where we're indicating that with the red is the GTP bound beta tubulin. When they're bound to GTP, they will associate with each other. They have a high affinity for each other, and they'll grow, right? They will be adding one at a time to each of those protofilaments, and over time it gets longer and longer and longer. We call this the GTP cap. But the tubulin dimer has intrinsic GTPase activity, which means once it's been assembled, at some point, the GTP that's bound to this green beta tubulin will be hydrolyzed to GDP. So everything in green is GDP tubulin. I think I've added that. no, I didn't. This in green is GDP bound tubulin. Red is GTP bound tubulin. And so if there's a high enough concentration of GTP bound tubulin dimers in the cytoplasm, then they will keep adding and our microtubule will be growing at its plus end. Right? So when we have enough GTP bound tubulins, we say that this is our GTP cap. But if the environment changes or under certain conditions, oh, there's our GDP tubulin in green. It's GDP, red, GTP bound. So GTP bound have high affinity for other tubulin heterodimers. GDP does not. Well, what kind of effect does that have? If this microtubule is in an environment where there's a very low concentration of GTP bound tubulin heterodimers, then all of the tubulins are bound to GDP. And when they're bound to GDP, they have less affinity for each other and they start falling apart. It starts peeling away, in individual heterodimers are then released into the cytoplasm as what we call free tubulin. As they lose affinity for each other, the microtubule will shrink. And this happens all the time to microtubules. Most microtubules are constantly growing and shrinking and growing and shrinking as what we call having a high degree of dynamic. They're right, microtubule dynamics, they're constantly changing their size. They're growing and shrinking. Now, they may be interacting with other proteins at this end that may prevent the disassembly of microtubules. There may be other proteins that will help bring in more GTP bound or help with affinity of GTP bound. But about 50% of all tubulin heterodimers are present in this pool. 50% of all tubulin is present as free tubulin in the cytoplasm. Free tubulin can exchange its GDP for GTP, and more GTP bound tubulin can then add to a plus end and provide another GTP cap, allowing it to grow. So there's always a source, there's always a pool of free tubulin that can be added, can allow for growth, but the concentration of that will determine whether or not any given microtubule is growing or shrinking. So we can see that here actually. So a quick little video right here. Let me close this down for a second. Go to a different part. There's a lot of really nice videos in SmartWorks here for us. And I could change my audio again.
All right, let's try it.
Microtubules continually grow from the centrosome added to a cell extract. Quite suddenly, however, some microtubules stop growing and then shrink back rapidly, a behavior called dynamic instability.
Oh, that wasn't the one I wanted to show. That's really boring. Sorry, let me try another one. This is the wrong one. I wanted to show you, this one. This one's much better. That was cute, but this is more interesting.
EB1 is a protein that binds to the GTP tubulin cap at the growing ends of microtubules. cells expressing a GFP-EB1 fusion protein reveal the spectacular dynamics of the microtubule cytoskeleton. Note that many, but not all, microtubules in this cell grow from the centrosome. Only the ends of growing microtubules are visible in this experiment. Those that are static or shrinking have lost their GTP tubulin caps and do not bind EB1. In contrast, when all microtubules are labeled with GFP tubulin, the true extent of the microtubule cytoskeleton emerges. Both growing and shrinking microtubules can be observed.
Okay, now that one was much more interesting, right? All right, let's go back. So
what they're showing us is, again, how quickly and why, not so much why, but the fact that the microtubules do change their length, right? They're dynamically unstable. This would be important in cell division. We'll talk about it in just a second, but let's ask a question about microtubule growing and shrinkage. Which of these situations listed here would enhance the shrinkage of a microtubule? So think about what causes microtubules to grow, what causes them to shrink, what's going to enhance their shrinkage. How about 5 more seconds? All right, I'm going to close it up. We've got a lot of people not voting. The only situation that would enhance shrinkage would be B. If you add more GTP bound tubulin, it will be able to grow, right? You'll have a GTP cap. If you inhibit GTP hydrolysis, then you'll never have any GDP bound tubulins. All the GTP bound tubulin dimers will have high affinity for each other. You'll maintain that GTP cap. If you increase the affinity of tubulin molecules for even the ones that are bound to GDP, if you increase their affinity for each other, they will stay together. Only if you inhibit the exchange of GDP for GTP, if you essentially are reducing the amount of GTP bound tubulin dimers in the cytoplasm, then there won't be any subunits that can add onto that plus end. Okay. Someone's asking about about static. Yes, and as a matter of fact, there are some tubulin molecules that don't grow or shrink. They are static, and that's part of their role. They have to remain one length. But first, before we go there, let's remember, why is that so important for these microtubules to have this dynamic characteristic? During cell division, our centrosome, also called MTOC, microtubule organizing center, will be replicated during interphase, same time that all the DNA is being replicated. Each centrosome is at a pole. The microtubules will start growing from the centrosome. Growing, the plus end will be towards the midline. All of those microtubules will then interact with chromosomes that they're not showing us here, right, at the midline. And then the microtubules have to shrink. They have to depolymerize. They have to shrink in order to pull that sister chromatid, one to the right and the other sister gets pulled to the left, right, to distribute that duplicated genetic information to both daughter cells. This behavior of microtubules is the target of some drugs that are used to try to specifically, or maybe not specifically, but preferentially damage cancer. Cells that are overproliferating, that are dividing too frequently, will be treated with chemotherapy drugs like vinca alkaloids, which are derived from a particular type of plant. These alkaloids will bind free tubulin, preventing that tubulin from being available to polymerize, to be added to a plus end of a microtubule, preventing the polymerization of microtubules. And so a cell that's been treated with a vinca alkaloid will be stuck sort of maybe in prophase or prometaphase. The microtubules can't grow towards the centromeres of the chromosomes. Another type of chemotherapy drug called a taxane, derived from a yew tree, will bind the microtubules themselves and enhance their stability, right? Maintain that GTP cap so that they can't shrink. If they can't depolymerize, then the microtubules can't drag the sister chromatids to opposite poles of that dividing cell. Either way, preventing polymerization of microtubules or preventing depolymerization of microtubules will prevent those cells from dividing. Right now, of course, these drugs will prevent any cell that's dividing or any cell that's trying to divide from completing cell division, but will preferentially be affecting the cells that are dividing very rapidly, right? Prevent their cell division. All right, we have to stop here. What we're going to look at next, and we'll start on Monday, is looking at some microtubules that are stabilized, some microtubules that do not continue to grow and shrink, and their role is to maintain a stable length. And we'll pick up there on Monday. Okay, anyone who has SmartWorks, take a peek at all of those videos. I'll highlight a couple of them on Monday as well, but they've got a lot of really nice visuals to help you, see and think, especially for dynamic changes in a cell. Have a good weekend, and we'll see you on Monday.